So it's my pleasure to introduce our third speaker, our third invited speaker, Dr. Georgios Nikolopoulos, who is actually speaking to us live from the conference room. Dr. Nikolopoulos is an accomplished epidemiologist, visiting assistant professor at the UCY Medical School, and he holds a doctoral degree in epidemiology from the Department of Hygiene, Epidemiology, and Medical Statistics of the Medical School of the National and Kapodistrian University of Athens, Greece. He's fellow member of the American College of Epidemiology, FACE, and certified in public health by the US National Board of Public Health Examiners. He worked as an expert in epidemiology at the Greek Center for Disease Control and Prevention for nearly 15 years, with involvement in the epidemiology and prevention of HIV, and in public health responses against SARS, West Nile virus infection, and pandemic A, H1N1 influenza. He was site PI for a US NIH study on HIV prevention and is currently leading a study in Cyprus on environmental epidemiology that is funded by the Cyprus Research and Innovation Foundation. We all know Dr. Nikolopoulos very well in his other role also as member of the scientific committee advising the Minister of Health on issues related to the COVID-19 pandemic. He used to be regularly on our television screens every Thursday, giving us all the statistics on the pandemic during the lockdown period, and we really appreciate him for his contribution to fighting the pandemic in Cyprus. So the floor is yours, Dr. Nikolopoulos.
So thank you very much for the nice introduction. I would like also to thank the organizers for this invitation, and especially my friend and colleague, Professor Fofi Konstantinidou. So my talk today will be about pandemics and public health. I will try over the next minutes to discuss with you about the history of pandemics and what we have learned over centuries, and some interesting epidemiological patterns. And then I'm gonna discuss about the components of our response to pandemics that include vaccines, surveillance, risk communication, and finally, I'm gonna touch a bit the problem with conspiracy theories and the anti-vaccination movement, which are problems we are likely to encounter over the next months. So, pandemic and epidemic, Greek words about problems in the population, pandemic about all people on Earth, and these usually refer to communicable diseases. Human history is full of outbreak events, of serious outbreak events, starting from ancient times. Here you can see the face of a young girl. She was probably 11 years old. The face was reconstructed based on a skull that was found in a mass grave in Keramikos in Athens, and she was probably a victim of the plague of Athens, an unknown pathogen, probably typhus or typhoid fever. And it occurred in Athens during the Peloponnese War with Spartans. A lot of people were gathered there, poor hygiene conditions, and many people died. And also Pericles, the leader, lost his life. And one lesson we can learn from the plague of Athens is that these major outbreak events may result in destruction of social fabric, and also perhaps it was a determinant of the imbalance, the geopolitical imbalance in ancient times between Athens and Sparta. Of course, plague is a term we use to describe a disease by another bacterium, Yersinia pestis. This is a very contagious and fatal disease, and there were some major outbreaks in human history. One of them in Roman Empire, another one more recently in China and in India. But what we probably know is the Black Death that occurred in 14th century in Europe. The name refers to the color of the skin due to necrosis and the sepsis of people who lost their lives. And what we can keep from this pandemic is first that approximately 1/3 of the population in Europe died. And this was a major event that changed the demography in Europe. And the second is quarantine. Now, most of us are familiar with this term. And it started during the Black Death pandemic. The word in Italian means 40 days. And this was a requirement for ships when they were coming to Venice to stay ashore to prevent the spread of disease in coastal cities. Of course, now plague has been considerably contained, and there are only a few cases worldwide because we have good treatment, we have vaccines, but however, there is still panic and fear associated with outbreaks. And this is a case in India in 1994. There were a few cases and some deaths from plague, but a lot of panic, a lot of fear. There were travel restrictions from other countries to India. And unfortunately, sorry. Το είχα κάνει. Yellow.
Okay.
Okay. As I told you, there was an event in India with few cases, few deaths, but a lot of panic and fear, restrictions, travel restrictions related to India. And as you can see in this photo here, a lot of people left the city of Surat, approximately 300,000 people in 1 or 2 days. And this was a lesson about outbreaks learned by the World Health Organization, and explains why the World Health Organization is very careful when it decides about travel restrictions, because they want to avoid overreactions to outbreaks. Cholera, another pathogen that has caused pandemics throughout the human history. It causes a watery diarrhea, which, however, could be very severe, and some people lose their lives. There were 7 cholera pandemics. One of them started recently, 1961, mostly in other developed countries. It later spread in 1971 in Africa, in 1991 in South America. And compared to plague, cholera still claims lives in other developed and poor countries, more than 2-3 million cases every year, and more than 30,000 deaths. However, I would say that in the 20th century, there has been a great progress related to infectious diseases. There were improvements in sanitation, better hygiene, vaccines, antibiotics, better medical treatment. And because of that, there was a great reduction in the incidence and deaths of infectious diseases. And this made the US Surgeon General in 1967 to say that it's time to close the book on infectious diseases. And this statement had a great impact on physicians, scientists, and epidemiologists who started working on chronic non-communicable diseases. However, he was wrong, because in the early 80s, we saw again an increase in incidence and mortality of a new disease. This is HIV/AIDS. We are probably, most of us, familiar with it. It started in early 80s. It took, however, and this is a difference between HIV and SARS-CoV-2, 2 years to discover the causative agent, which was a virus. Unfortunately, we have not found a vaccine despite the efforts, and almost 40 million people have died since the early 80s. There are a lot of lessons we've learned from the HIV/AIDS pandemic, and one of them refers to stigma and discrimination for certain population groups, including the gay community, people who inject drugs, sex workers. However, and this was something really good, these people came together and managed to put HIV/AIDS in the research agenda. And as a matter of fact, one of the people from the scientific community who was actively involved is Dr. Anthony Fauci, who is well known today for dealing with the pandemic. And because of the struggle of these people, and of course, scientific research and progress, we have now very good treatment, and people live as long as people without HIV. And in addition, treatment can be used as a tool for prevention. And nowadays, we can be very optimistic that in 10 years from now, it is likely to put an end to HIV pandemic. We are not going to eliminate the disease, but it won't be a major public health threat. Of course, when we talk about pandemics, we think about influenza. This is a viral disease. It is spread from person to person through respiratory secretions. We are all familiar with seasonal flu, which comes from one winter to the next winter. Some people become sick because the virus every year is slightly different. However, sometimes because of events that relate to exchange of genetic material between viruses that infect different species, domestic birds, pigs, and humans, a new pathogen emerges with a completely new genetic composition, and most people are susceptible to become infected. And then a pandemic may start, especially if the new pathogen is able to infect humans, to reproduce efficiently in humans, and also to be able to spread from one person to another easily. And human history, again, has experienced a lot of pandemics from flu strains, some of them in the 20th century and one recently in 2009. Let's discuss some things we have learned about the epidemiology of those pandemics, which could be useful for our effort today with COVID-19. You can see that, masks were something people used to fight the pandemic, social distancing measures. However, they were not universal. And of course, this pandemic hit hard the human population. As you can see, approximately 1/3 of the population was infected. 1 out of 10 people of those infected died, a huge fatality rate. And it is estimated that more than 50 million people died, which is approximately 2.5% of the total population on Earth in 1918. There are two interesting things about the flu pandemic in 1918. The first is the waves, and that's why we're discussing now about a potential second wave of COVID-19. The first was very small. However, the second, in the fall of 1918, was huge, very devastating, and this was later followed by a third wave. So what we know from the history of pandemics is this, and especially of influenza, it may come in successive waves of different size and severity. And the second thing about the pandemic of 1918 is that it hit hard young persons. You can see here the shape of mortality curve, which is usually U-shaped in human populations. However, this changed and became W-shaped with the peak corresponding to the high number of fatalities in those age between 25 and 45. And you can imagine how important and the impact on societal and economic productivity if young people die. There were also two other pandemics later in the 20th century, one in 1957. As you can see, this actually consisted of one big wave. The fatality was smaller and it killed approximately 1 million people. And another one 10 years later with two waves, a small one followed by a big one 1 year later. And again, the fatality was relatively small compared to Spanish flu, but it's still, when this small proportion refers to a totally susceptible population, the absolute number becomes very, very high. Certainly, we remember, I think, the recent pandemic influenza of 2009. You know, the presidents of the United States are always in the epicenter of interest. Then was Barack Obama. The characteristic of pandemics, of flu pandemics, is that they can spread very, very fast. As you can see here, the first cases of increased activity of respiratory infections was detected in Mexico in mid-March, and I remember it very well because I was then at the Greek Public Health Agency and I drafted the first report that this was strange to see an activity in March in the Northern Hemisphere. And in 1 month, there was spread in other continents as well. And in June 2009, in early June, the World Health Organization declared a pandemic. And I am mentioning this because there was some discussion now with COVID-19 whether WHO was late or the reaction was early. I believe that it was quite on time because it could not be earlier. And I mentioned the example with the plague outbreak in India, which had enormous consequences in economic terms. And it was not justified by the size of the outbreak. On the other hand, it cannot be very late. So it takes 2 or 3 months to evaluate the data and decide whether this is a serious public health emergency that requires the mobilization of human and material resources throughout the globe. Certainly this pandemic, although it lasted more than a year, so when pandemics come, we have to be prepared for that. It takes time to come to an end. You can see the fatalities, the recorded fatalities were less, although there are estimates from models that it was perhaps about 300,000 or 400,000, but still the fatality was small, and the WHO evaluated it as a moderate pandemic in terms of severity. But certainly, this pandemic also came in 2 waves, a very small wave in spring, summer, followed by a big wave in the fall. So given this history about pandemics, we now are experiencing COVID-19 pandemic. It started with a few cases in China. We are all now aware of it. At the beginning, there were some concerns in major journals, scientific and media. I must admit that I underestimated the threat because I remember that I was involved when I was in Greece in coping with SARS-CoV-1 epidemic, which proved to be minor. It was contained in a matter of months. However, this proved to be different. There was a potential for a pandemic, so there was spread in other continents by late January, and in late March spread worldwide. And you are now aware of the fact that more than 1 million people died and more than 30 million people have been infected. In terms of what we know about the new disease, it is a virus. We call it coronavirus because it looks like a crown under the microscope. with these surface proteins, the spike protein that is used to attach to human cells and enter in the human cell. This is not a unique pathogen. I mean, there are other coronaviruses that circulate in humans. Two of them, you can see that they are very related to SARS-CoV-2, is the SARS-CoV-1 that started in 2002, 2003, and was easily contained, MERS, which emerged in Middle East in 2012 and still is circulating, but it is very controlled. But you can see with the red color some other coronaviruses that circulate in humans and cause mild infections. However, it is believed that perhaps some of them were also cross-species transmissions that resulted in some outbreaks that we were not able to detect. And there is some theory that one of them was actually the cause of the Russian flu in 1890, that this was not actually a flu, but a coronavirus. In terms of transmission, there is person-to-person transmission through big droplets from one person to another. However, it seems, unfortunately, that there is also aerosol transmission, which means that there are some particles very small in diameter that stay in the air because of their size. They don't go down, so they stay in the air for a while, and they may circulate in a longer distance, and people, especially in close, crowded areas, may inhale the pathogen. But certainly the transmission through droplets is the major route, and also it seems that it could be, or it has been transmitted through touching contaminated surfaces and then putting the hands in the face. Now, in terms of its fatality, certainly it is not SARS-CoV-1 or MERS, which were very fatal. It is estimated that it kills between 0.1% to 1% of people who are infected. It seems low. 0.6%, but if you this results in absolute numbers in a lot of fatalities. It is certainly contagious. The reproductive number is 2.5. Automatic transmission, and now there is a lot of research to understand whether people produce antibodies, what type of antibodies, what type of other cell responses. This is an unknown, and there is a lot of research about it. And closing this first part, pandemics do not produce only curves of cases and deaths. We may have curves of recession. They have economic consequences, and governments try to flatten not only the real epidemic curve, but also the recession curve. And one not unwanted, actually something nice, but it happened during a pandemic, that because people stayed at home, there was a reduction in fossil fuel consumption. And you can see this reverse curve again that started in China and then spread to other countries. Now, in terms of responses, of course, we all know about vaccines. There are also, however, other important components of responses. One of them is surveillance. It is important to diagnose the problem, and the surveillance means established mechanisms to collect information. This information should go to public health authorities to analyze it, to interpret it, and then disseminate these findings to people who should take decisions. And the intervention should be based on this data, and we anticipate changes. And I think this is in the core of our response, because if you don't understand your epidemic, your pandemic, your intervention might not be effective or efficient. And surveillance is more complex than what most people think, because usually we listen to the news that, okay, today 10 cases, 20 cases. This is not enough. I mean, we have to take into account the size of the population, and also in order to study the disease, to think about all the different phases in which people may be during their illness. So some people are asymptomatic. We need seroprevalence studies to estimate the proportion of people with antibodies. Some people are symptomatic but do not visit the physician. There are some nice ways to get information by looking in Google searches, and actually one colleague of mine will later present some of these efforts. Some people go to the primary care physicians. This is another thing we must capture to see how many people visit primary care physicians and what proportion of them is due to COVID. And then people may go, if the disease is more severe, to hospitals. It is important to capture the number of admissions, how many of them were due to COVID, and of course, some hard outcomes like admissions to intensive care units and deaths. And I believe this should be the outcomes to evaluate our response, I mean epidemiological outcomes, when this epidemic finishes. Of course, the most important component of response is our own immunity, our own immune system. And let me tell you, before going to vaccines, a few things about immunity. There are cells in our body that bind to pathogens when we are infected and present some parts of the pathogens to other cells, either B cells or T cells. When B cells are activated, they produce antibodies. Antibodies bind to proteins, to viruses, and these complexes later are destroyed. And also there is a T cell mechanism, another way to destroy, this time, not viruses, but cells that contain viruses. And when we are talking about immunity to COVID-19, it is very important if vaccines, for example, can generate both approaches, both mechanisms of response, both B cell and T cells. Of course, the most effective way to deal with an infectious disease is to produce a vaccine. And vaccines are biological material that is used to train our immune system to fight pathogens. And the administration of vaccines is known as vaccination. The name vaccine comes from the Latin word vaxa, which means cow. And this relates to the father of vaccine, Edward Jenner, who used pus from pustules from people who were infected by cowpox and injected it the first time in a small boy in England in 1796, I think. And by doing so, this boy was protected afterwards by infection of smallpox. And smallpox, I'm going to talk about it later, was a very serious, contagious, and fatal disease throughout human history. From this first effort, now we have a lot of nice technologies, some of them very good and safe, like nucleic acid-based vaccines or vaccines using vectors. I'm going to talk about two of them because actually these vaccines are in advanced stages of development. One of them is based on mRNA. What they do is that they get a genetic sequence, a small genetic sequence that, if it is translated, results in the production of the protein, the spike protein of SARS-CoV-2. They put it in a lipid coat. It is injected. It is then absorbed by the cells, and then the cell machinery uses this information to produce proteins of SARS-CoV-2. A very safe way to do that, to trigger and train the immune system. The second, and this is the platform used also by AstraZeneca and Oxford, is based on vectors. Vectors are mild viruses, very mild viruses that are harmless to people. Actually, they have been changed to lose reproduction properties, replication properties, and also they are genetically engineered to insert a part of the genome of SARS-CoV-2, and then a lot of viruses, a lot of these vectors are injected, billions of viruses. They go into the cells. As I told you, they are harmless, but the genetic information they include is translated, proteins of SARS-CoV-2 are produced, and the production of antibodies starts. Of course, vaccines are very important because they do not only protect people who are vaccinated, but they create also collective herd immunity. So if we are vaccinated, we may protect some other people who did not get the vaccine for various reasons. For example, small children, people with compromised immune system, and etc. So this is also another important property of vaccines. Because of vaccination, in the 20th century, we managed in developed countries to decrease the incidence and mortality of a lot of pathogens, a lot of infectious diseases. And perhaps the major public health achievement in human history was the eradication of smallpox. As I told you, it was very contagious, fatal, and even people who survived smallpox, they had scars in their faces, some were blind, and it is estimated that 2 to 3 million people were dying each year in the 20th century. However, there was a good vaccine based on the work of Edward Jenner, and in addition, it was also an advantage for humanity that this virus was only found in humans. There was no animal reservoir. So WHO decided to implement a plan to eradicate the disease. And this plan was based on two important components. One was vaccine, the other was surveillance. So what they did was to massively vaccinate people. Of course, herd immunity was reached, but still there were some local cases of the disease. That's why they developed good surveillance system in poor, underdeveloped countries, but they were very effective. And by doing so, as you can see to the left on the slide, they were able to find cases, and then they did contact tracing. I think we are all familiar with that now. There was a first cycle, a first wave of contact tracing, and then a second contact tracing of the contacts of the case. This is known as ring vaccination, and by doing so, in 10 years, they managed to eradicate the disease. The last case was in 1977, and officially in 1980, eradication was eradicated from the planet. Nowadays, in terms of SARS-CoV-2, there are many vaccines in phase 3 trials. As you can see, some of them based on vectors, others on mRNA or protein-based, some based on inactivated viruses. Major work in the United States, in China, in Russia, in the UK. So we can be very optimistic that probably by the end of this year, one or more vaccines will get at least an emergency use authorization. Some people raise arguments here that, okay, is it possible to develop a vaccine in such short period of time? Because we know vaccines take long to be developed. However, this time things were different. First of all, there is an improvement, scientific progress. Remember what I told you about HIV? It took 2 years to detect HIV, but only a month to identify SARS-CoV-2. In addition, there has been a lot of work in the preclinical stage. So we know some things, the scientists, vaccinologists knew a lot about work with animals, previous work with SARS-CoV-1 or MERS, and a lot of work with adenovirus in other infectious diseases. So it was easy to go fast to human trials. And again, in human trials, some phases proceed in parallel, not in series. There was less bureaucracy. Perhaps we have heard that the EMA has started of vaccines before formal application to speed up the process. And also something very important, as you can see on part A, after approval, the production starts. However, now governments took the financial risk and supported the manufacturers, and they started producing the vaccines before approval. So if they prove to be not safe or not effective, there is going to be a financial risk. If, however, they are safe and effective, they will be available pretty soon. And finally, certainly beyond vaccines, we need better treatments or better therapeutic approaches. And now let's see a few things about communicating risk and prevention messages, which are also important. I wouldn't say that I am an expert in this, but based on my experience, I think that it is an important component of responses to outbreaks and pandemics. Communicating risk in pandemics means to exchange information between experts and people in order people to make informed decisions. Certainly, communicating risks and convincing people of a public health threat should not be like some approaches in Indonesia, where people who did not wear masks were forced to dig the graves of people who died from COVID-19, or some others were put in coffins because they did not wear masks. And certainly, even if there are not these, let's say, hard approaches, we should be careful not to distract people from the essence, the core of a message. In Greece, they decided to distribute masks to young children, but unfortunately, when they did that, the size of the masks was huge. Actually, it was a full face covering. And this was bad, I mean, when you communicate prevention messages. If there are some principles in communicating risk, based on my experience and not my expertise in this field, I would say that it is important to brief media regularly, to disclose promptly updated information, to build trust, to listen and understand people's beliefs, to put credible people to give advice. Very important to engage community, to be honest, to avoid absolutes in public health. In some cases, there is no black and white, and also that science is uncertain, and we may change messages like we did with face masks. And something very important for me as a person, and based on my experience with HIV, we should show that we care for people, that there are not cases of the disease, but people with COVID-19, and also to avoid stigmatization of any kind. And if there is an effective risk communication, it is also likely to be able to manage rumors, misinformation, and stigma on time. Now, of course, after 6-7 months in the pandemic, there are issues, pandemic fatigue. It is recognized by WHO. So we should take this into account when we plan responses. It is important to consult with communities to understand why they are frustrated, to see what kind of policy will work at the end, and to balance scientific, social, and political needs. To my opinion, it is also important in advisory committees to include not only clinicians and epidemiologists. This is not a medical problem. This is a public health, societal, economical problem. So it is important to get involved experts in communication and, of course, psychologists and sociologists. And we should also remember that even members in advisory committees become fatigued, and not only here in Cyprus, but everywhere. And finally, I would like to touch shortly the problem with the anti-vaccination movement, because it is likely to face it in the next months as vaccines will become available. And of course, the problem with conspiracy theories. This is something I know well from the field of HIV, in which I have long-term experience. I remember even when I was a teenager in the 80s that there were discussions about a man-made virus of President Reagan to kill gay men in the United States. Of course, it took some time to science to prove that this was wrong. Now we have very good data that this was actually, again, a cross-species infection from chimpanzees, monkeys in the early 20th century. It was a pathogen circulating in humans for many decades, so it couldn't be something made in laboratories. Unfortunately, even now in the United States, among certain communities like Afro-American men who have sex with men, they still believe, 50%, that this was a man-made virus about HIV. In COVID-19 era, we have new conspiracy theories, pseudoscience spreading fast in social media, and of course the anti-vaccination movement, which is somehow related to conspiracy theories, but it is not something new. It has its roots in the first years of vaccination, and actually this is a caricature of 1802 making laugh at the Edward Jenner's vaccine, that people, if they get a vaccine from cowpox, they will have bovine figures on their arms, hands, and faces. In this photo, you can see demonstrations against compulsory vaccination in 1918. And if we read the history of vaccination, we will see that the first event that created resistance to vaccination was the Compulsory Vaccination Act in England in the mid-19th century. And unfortunately, later, some events in the 20th century, some lots of polio vaccine that the virus was not inactivated, the unfortunate event in 1976 with swine flu, some rare adverse events with pertussis vaccine, which was very, very effective and protected children. The death rate of pertussis was more than 10%, but unfortunately, these rare events, which actually were not associated with the vaccine, changed the attitudes of parents, and there was a decrease in vaccine uptake in the 70s and 80s. But perhaps the most influential paper was that appearing in Lancet in 1998 by a physician named Wakefield, who, using 12 children, he claimed that there was an association between the vaccine for measles, mumps, and rubella with regression in children's development and also some bowel problems. Although this paper was retracted for various reasons, for misconduct, combating interest of the first author, it remained influential. And despite the fact that there have been many studies with 1,000 children in good papers which showed that there was no association. In any way, I think that it is very important to understand the problem, and here in Cyprus as well, at least to see if there is a problem, because there are multiple reasons for these attitudes, not only conspirational thinking, but lack of knowledge, perceptions, religious belief, political beliefs, and sometimes even geopolitical tensions. in 2012, CIA in Pakistan organized a fake vaccination program for hepatitis B to get samples from people there in order to identify Obama bin Laden and his family. This operation was effective, but unfortunately, it destroyed for some years the efforts to vaccinate people for polio in the region. For COVID-19, there are some polls which say that in some countries, 1/3 of people are not going to take the vaccine. This is not good, and certainly if it is combined with misinformation in the media, may result in catastrophe, in a public health catastrophe. I remember something from the 2009 pandemic. We wrote some letters in public health journals. In short, here you can see the uptake, the vaccine uptake of 2009 vaccine, especially among the general population. There was a TV show which showed an association in a popular TV show in Greece, an association between a woman who had a tremor in his head and the vaccine, which proved to be wrong, of course, but in 1 or 2 days, the vaccination uptake went down. And unfortunately, and we wrote it in another paper, that the next year, when the pandemic, the 2009 pandemic became seasonal, more people died because they did not get the vaccine. Because there are studies which show if you get the vaccine 1 year, it is very likely to get it the next year. People did not take it, so it was more likely not to take it the next year as well. In terms of how to deal with vaccination, it is important to find modern communication solutions, to build trust, to engage community, all stakeholders, to be proactive, to send mass communication messages, and in addition to perhaps if there are conspiracies who stop to be members of this group, to bring them on the surface and discuss what the group's beliefs are and try to persuade other people, and of course collaboration with social media platforms which could proactively monitor, flag, and remove harmful health information. And closing, I would say that we should remember that pandemics of communicable diseases are part of human history. We have to live with that. Of course, these pandemics have serious public health consequences, but also societal and economic consequences. And that's why it is important to have a holistic approach. In the early days of pandemics, there are uncertainties, and we must learn to communicate these uncertainties to people. It is important to involve the community, to build trust with the community. If we lose this, it is highly unlikely to gain trust again. Interventions, including vaccines, should be based on science. But in addition, it is very important to understand your pandemic. Your responses should be driven by the diagnosis you make. It is important to know the epidemic. Perhaps something that works here in Cyprus does not work in Greece or in another setting. But certainly, given the progress we have seen, we should be very optimistic that we will survive this pandemic as well. Thanks a lot for your attention.
So that maybe I'll start the video as well. So one question from our Facebook audience: how dangerous is it to give drugs which have not been adequately tested? For example, with regards to President Trump, where pharmaceutical companies may also be under pressure to produce before proper testing is completed.
Okay, so we are talking about therapeutic medicines. Although I am not completely aware of what President Trump actually was given, I think two of the medicines that he took are approved for COVID-19. One is remdesivir and the other is dexamethasone. The only investigational drug that he took was monoclonal antibodies. This was, I think, in phase 3 trials, and probably manufacturer applied for emergency use authorization yesterday. So even for President Trump and for everyone, sometimes if a medicine seems promising without approval, and given that there is nothing else to give to a patient, a drug without approval may be given. But certainly when there is any now, there are at least 2 drugs that in clinical trials have been shown to have some effect. As I told you, remdesivir and dexamethasone. And this is, as I said, the good way to go. I mean, to be based on science in order to administer the drugs.
Thank you. Another question from colleague George Panoudis. You said that soon we will have vaccines, but in fact we have one, the Russian vaccine. What is your comment on the Russian vaccine?
The Russian vaccine. The Russian vaccine was in phase 2 trial. Now it will enter phase 3 trial. Remember what I told you about, and probably you are well aware of clinical trials, that phase 1 is important to see whether a vaccine is safe or not. Phase 2 to see if there is immunogenicity, that humans produce antibodies. And then in phase 3 trials, large groups of people are compared. One group that takes the vaccine and another group that takes the placebo. And this is the most important part of a trial. In Western countries, and I think in developed world or everywhere, before applying for emergency use authorization or for regular approval, you have to show data that your vaccine works in phase 3 trial. So probably what happened in Russia is that there was an approval to enter a phase 3 trial. So we do not have data from phase 3 trial from the Russian vaccine. In terms of the vaccine per se, it is based on vectors. It is a clever, a smart idea, I would say, because it combines 2 vectors. They are human vectors, different from the Oxford vaccine, which is based on chimp viruses. One is given initially and the second 1 month later. And why it is a smart approach? Because unfortunately, these viruses, adenoviruses, produce common cold, and a lot of us may have antibodies against them. So the vaccines actually may not work because of that. So by doing so, having a prime boosting with one vector and then with the other vector, they may overcome the problem. But again, it is not a fully evaluated vaccine. It now enters phase 3 trials, and we will be waiting for the results. Some other vaccines, including the vaccine of AstraZeneca, of Pfizer, which is an mRNA vaccine, and Moderna, it is likely to have results, I think, by late November or early December. Not approval, results.
One more question is, what is the false positive rate of the tests, diagnostic tests?
Yes. I think the diagnostic test that we use, although I am not an expert in testing, is PCR. So it tries to detect the genome of the virus. As with all diagnostic testings, we require both high sensitivity and specificity. And I think especially for PCR, this is in the level of 98%, 99%. So they are generally very accurate tests to detect infection. I think it is important, however, to make sure that the lab and the lab people are experienced with this technique, and to avoid potential lab errors or errors in sampling that they may result in wrong results. In general, however, I would say, and especially about Cyprus, that there has been a process to make sure that the quality of labs that perform PCR testing in Cyprus is good. And despite some errors, a few errors that were very early detected, we have a very good picture of what is happening, both at the individual level, I mean about diagnosis, and the population level. So in general, people should trust the PCR testing, and especially PCR testing here in Cyprus.
Questions. I'm going to combine the next one with one of my questions. You mentioned earlier the need for the involvement of other scientists, psychologists and sociologists in the national campaigns of different countries. We know here, and we heard from our previous speakers about the involvement of psychologists in other countries, which is very intensive. This is mainly a behavioral problem at this stage of the pandemic. So, do you think you should involve or the government should involve experts in the human behavior sciences in the next steps of the pandemic here in Cyprus as well?
Yes, and it is clear, it is crystal clear that I believe not only behavioral scientists, I mean from the psychology field, it is both a behavioral and societal problem. So we need both psychologists and sociologists. And especially given the need to vaccinate people, it is very important to understand here in Cyprus whether there is an intention to get the vaccine, what is the proportion of this intention, and what are the determinants of this intention. And if there is a low level of people who want to get vaccinated, to see if these determinants can be changed. So perhaps, although I believe that these scientists should have been involved in this process, perhaps now it is more important to bring them in the team.
Yeah. Okay. Let me see. One more question that's related to what you just said. Do you think that vaccination should be done on a voluntary basis, or are there enough reasons based on the economic impact that it should be mandatory?
No, mandatory, and unfortunately, history gave us a lesson, because they tried to make it mandatory in the mid-18th century. And there was, first of all, a resistance by the working class then in England, who were feeling oppressed and thought of it as a new method of oppression. So there is a long history with trying to make vaccination compulsory. I disagree with that. We should do it on a voluntary basis. The important thing to do, however, is to understand what are the drivers, the determinants of vaccination, to address them, to inform people, and perhaps to those who select to get the vaccine, to give some motives. So instead of punishing those who refuse vaccination, to find other ways to motivate people to get the vaccine. So certainly, I don't think a mandatory approach would help. Instead, we should find, modern and smart ways to upscale vaccination in Cyprus and everywhere.
Okay. And research in Cyprus shows, as we all see around us, that certain groups of people demonstrate low adherence to precautionary measures Will you use the information available to advise the government for creating targeted campaigns?
Say it again, sorry.
Yes,
Yeah.
maybe I'll take the mask off.
Oh.
We've observed through research, this is a question from Fofi, from research here in Cyprus, that there are certain groups of people demonstrating low adherence to the precautionary measures. Will you use the information to advise the government for creating targeted campaigns?
Yes, certainly, and we need the research in this field. And I told you that all interventions, not only biomedical interventions, I mean behavioral approach, societal approaches need data from good scientific work. So we need this information. Otherwise, unfortunately, our approach will not be efficient. In the first wave of the pandemic, it was easy to select some strict measures, to ask people to stay at home, and actually this was enforced. And we know it is very effective, but certainly it cannot last, and given the pandemic fatigue and perhaps cultural characteristics of Europeans, it is impossible maybe to follow this pattern again. So we need this information. We need to understand people and how to convince them to get involved. And you know, the lesson from other diseases like HIV is that your interventions will be more effective if you involve the community, if we involve people in this. It is very important.
I have one more question. I don't know if I have another one from the audience, but there's something I was curious about since the beginning of this pandemic. In the early days, when the virus was still contained in China, the World Health Organization made an announcement that it's not a good idea to stigmatize China and to close the borders of, outside of China. And this was when the virus was still contained. If the WHO had taken a different approach, since the borders closed anyway in a few weeks, and closed the borders of China early on, do you think it would have made a difference given the response of China now, or would we have had the same situation just delayed by a few weeks or a few months?
Yes, probably the latter. I think I mentioned at the beginning this outbreak event in India, and overreaction may harm certain regions. And this was included in the international health regulations. So there is a kind of legal framework how to react in outbreak events to avoid travel restrictions and stigmatizing regions. Perhaps I think it would be more helpful if we improve our surveillance systems. And maybe this went wrong in China. I mean, it is important to find very early these cases and do some good epidemiological work to understand early that this was not related only to the Wuhan market, but there was a person-to-person transmission. If this had been done, perhaps some local measures could be efficient to control the pandemic. By the time the WHO was informed, perhaps it was too late to contain it. And in general, my approach to this is to avoid stigmatization, to avoid this kind of measures. We live in a global community, and we have to balance this. Living in a global community means easier spread of pathogens. So perhaps better surveillance systems, and especially in this area of the world where new pathogens appear, would be the best way to go in the future.
Okay, thank you. We have to close. Thank you very much, Georgios. Very informative.